Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about construction of recombinant cDNA from an mRNA. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So, complementary DNA or cDNA is produced from mRNA. Now, clones of such DNA copies of mRNAs, that means clones of cDNA, are called cDNA clones. So, why do we prefer cDNA clones over genomic DNA clones? Actually, in eukaryotic cells, the mRNA is spliced before translation into protein. Because we know that when a native mRNA is produced after transcription of DNA, it contains both exons and introns. And introns are non-coding parts. So first, these introns are needed to be removed from mRNA. And then only the mRNA gets matured and it leads protein synthesis or translation. Now, the mature mRNA will produce cDNA. So, the cDNA synthesized from the spliced mRNA doesn't have any introns. Therefore, the protein to be expressed can be sequenced from the cDNA, which is the key advantage of cDNA cloning over genomic DNA cloning. Hence, we prefer cDNA cloning. Let's check the steps of constructing a recombinant cDNA from mRNA. So, we will see the diagram first. So, here we have our target cell. From this target cell, we want to extract mRNA or isolate mRNA. So, in this target cell, not only the mRNA, but tRNA, rRNA, all will be there. So, we can isolate the total RNA mixture from this cell. And from that mixture, we will get our mRNA only. So, we can treat these mixture with some reagents that will separate tRNA or rRNA and we will get our mRNA only. While we have our pure mRNA in our hand, what we can do? We can hybridize these mRNA with oligo-DT primer. Why? So, this is our mRNA. You can see the mRNA is obviously single-stranded and in this mRNA, its 3' end contains poly A tail. That is obvious in mRNA that we know that after processing, mRNA has poly A tail in its 3' end. Now, you don't know the sequence of these mRNA. So, what you can do, you can just hybridize this mRNA with oligo-DT primer. That means your primer will come here and will hybridize with this poly A tail of mRNA. So, this is done. Now, what we can do, we can add a special enzyme, reverse transcriptase enzyme. So, reverse transcriptase enzyme will continue reverse transcription. That means reverse transcription will happen. What does that mean? Actually, reverse transcription means that transcription is reversed. That means we can create DNA from mRNA. So, here reverse transcriptase enzyme will do the same. Now, this is the DNA part and this is the mRNA part. So, we call it mRNA cDNA hybrid. In this hybrid, one strand is of mRNA and another strand is of cDNA. So, this is the single-stranded cDNA and single-stranded mRNA hybrid. Now, we treat this hybrid with RNase H. So RNase H actually removes this mRNA and we will have the single-stranded cDNA only. So we have single-stranded cDNA in our hand now. Now what we will do, we will add poly-DG tail at 3' end. So we have added here poly-G tail at 3' end of this single-stranded cDNA. Why we will do that? Because we want to make it double-stranded. Now if you want to do that, you need primer. So again, your primer will be oligo-DC primer. So you can hybridize this single-stranded cDNA with oligo-DC primer. In order to put this oligo-DC primer, you have added this poly-DG tail at 3' end. Now you have kept your primer here. So your primer will hybridize with this. And now you add DNA polymerase. So DNA polymerase, we all know what does it do? It actually extends the strand and makes DNA. So this CCCC primer, this primer will be extended by DNA polymerase and it will produce double-stranded cDNA. So now in our hand we have double-stranded cDNA. Next, what we will do, actually we want to put this double-stranded cDNA in vector. So in order to do that, you have to make compatible sticky ends in both double-stranded cDNA as well as vector. So you can add a linker that contains a restriction site. In this case, we have added a linker that contains EcoR1 restriction site. So this is our linker. And this linker will be placed at both the ends of this double-stranded cDNA just like this. Now we will cleave this linker with EcoR1. So this EcoR1 actually cuts site after G. Like this. And we get these fragment. And here we have sticky ends. Now we will cleave the vectors also with the same restriction enzyme, that is EcoR1. It will make a free space in vector. Then we can just ligate these two. So we will mix this EcoR1 digested double-stranded cDNA as well as digested vector, and we will put the T4 DNA ligase. So it will add this double-stranded cDNA in the vector. Then we will simply transform the bacterial cells using this vector. Suppose in this case we have used plasmid, so it is easy to transform bacterial cells using this plasmid vector. And our transformation is done. So in our bacterial cells, now we have our recombinant plasmid vector, and that contains recombinant cDNA. Let's check the text once. So first step is isolation of mRNA. Now total mRNA is isolated from cells. So it is desirable to remove tRNA and rRNA. Obviously, because we want purest mRNA in order to do the downstream processes. Next is synthesis of the first strand of cDNA. So mRNA, being single-stranded, cannot be cloned because vectors are double-stranded, right? If we use plasmid vector, it is double-stranded. So you need a double-stranded cDNA. So it is first converted into DNA prior to insertion into a suitable vector. An oligo-DT primer is annealed to the poly-A tail on the mRNA. Then reverse transcriptase will begin DNA synthesis at the primer. That means reverse transcription will be done. The mRNA from the cDNA-mRNA hybrid can be removed by RNase H treatment to give a single-stranded cDNA molecule. Then poly-DG tail should be added at the 3' end of single-stranded DNA. Next is synthesis of the second strand of cDNA. So an oligo-DC primer is annealed to the poly-G tail on the single-stranded cDNA. The single-stranded cDNA is converted into double-stranded cDNA by DNA polymerase enzyme. Next is incorporation of cDNA into a vector. So in order to incorporate a cDNA into a vector, both need to generate compatible sticky ends. Linkers carrying a cleavage site for a restriction endonuclease can be ligated to both ends of cDNA as well as vectors. See, if the vector contains the same restriction site, then we don't need to add such linkers in the vectors. But if the vector we have chosen doesn't have the restriction endonuclease site that we want to be there, that time we can put the same linker in the vector so that it contains the same restriction site. Now restriction digested cDNA and vector are joined at the ends using DNA ligase to form recombinant cDNA vectors. Last process is transformation. The ligated cDNA vectors, here suppose we have taken plasmid, are then transformed into bacteria. And this is all about today's lecture. I hope you liked the lecture. If you want to get the PDF notes of this topic, please check the first pinned comment of the description box. Thank you.